Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Harris. I wanted to use office hours to talk about my paper on extremophiles and life detection for astrobiology.
Good afternoon, Maya. That is a rich topic. What aspect are you focusing on? Organisms in analog environments, or the instrumentation used to detect life signatures?
I want to connect both. I am trying to explain how studies of extremophiles on Earth inform the design of instruments that search for life on Mars and icy moons.
Excellent. Start by summarizing the kinds of extremophiles you plan to describe and why they matter for detection strategies.
I cover thermophiles from hydrothermal vents, psychrophiles from Antarctic brines, halophiles from salt lakes like Mono Lake, and acidophiles from places like Rio Tinto. I argue that each group shows different biosignatures, such as unique lipids, specific isotopic fractionation patterns, and molecular chirality.
That is a good range. Make sure you explain concrete examples where an extremophile yielded a biosignature that instruments could detect remotely or in situ.
For instance, lipid biomarkers from sulfate-reducing bacteria at hydrothermal vents show distinctive haploids that are stable in sediments. Also, amino acids from microbial mats often show a left-handed preference, which we can measure as enantiomeric excess.
Yes, chirality is an important biosignature, because many abiotic syntheses produce racemic mixtures without an enantiomeric excess. Mention that alongside isotopic fractionation of carbon and sulfur, because living metabolism often leaves measurable isotopic signatures.
I will. I also plan to discuss false positives. In the Atacama, abiotic organics persist and can mimic biological patterns if you only look at total organic carbon. That history shows why context is critical.
Good. You should discuss the Viking lander experiments and their ambiguous results. People still debate whether the labeled release experiment detected metabolism or an abiotic oxidant in the soil.
I mentioned Viking and then linked to modern instruments. For example, the sample analysis at Mars instrument on Curiosity uses gas chromatography mass spectrometry to analyze organics after pyrolysis. I explain its limitations with refractory organics and perchlorate interference.
That is a key point. Perchlorates can destroy organics during pyrolysis and create chlorinated hydrocarbons that are ambiguous. That is why complementary methods like Raman spectroscopy and laser desorption mass spectrometry are valuable.
I plan to compare techniques. GCMS is great for volatile organics and specific biomarkers. Raman gives mineral and organic structure without heating. Laser desorption can analyze complex organics without combustion.
Also mention nucleic acid-based methods for detecting extant life. PCR and sequencing work well in Earth labs, but they assume familiar genetic polymers. If extraterrestrial life uses different biochemistry, we may miss it.
That is one of my conclusions. We need both agnostic detectors that look for patterns like non-random elemental distributions, and targeted detectors that look for Earth-like biomolecules such as DNA or ATP.
Exactly. Give students an example of an agnostic approach, for example measuring chirality, isotopic fractionation, molecular complexity, and spatial pattern at microscales. Combining these can indicate biology without assuming a specific polymer.
I will show a case study from the dry valleys in Antarctica, where scanning electron microscopy and fluorescence microscopy revealed microstromatolites with isotopically light carbon and layered organic structures. That combination made a stronger case than any single test.
Good case study. Also discuss planetary protection and contamination control. Field studies of extremophiles taught us how easily Earth microbes hitchhike on spacecraft. Sterilization protocols are essential to avoid false positives.
I described sterilization, clean rooms, and the role of witness plates to monitor contamination. I also cover forward contamination and the ethical reasons to avoid introducing Earth life to other bodies.
Make sure you explain why multiple lines of evidence are necessary for declaring detection. A single organic molecule can be produced abiotically in a variety of chemical reactions.
So the conclusion will stress the need for mission designs that include orthogonal instruments. For example, a rover that carries a mass spectrometer, Raman spectrometer, a microfluidic immunoassay, and microscopes would be ideal to cross-validate results.
Yes, and discuss trade-offs. Instruments increase mass and power costs, so mission architects must prioritize. Your paper should analyze which biosignatures give the highest confidence per unit resource.
I was thinking of ranking biomarkers by robustness and detectability. For example, isotopic ratios and chirality are robust and not easily produced by simple abiotic processes. Intact nucleic acids are easy to interpret but degrade quickly and require assumptions.
That ranking will make your paper practical and not just theoretical. Also include suggestions for future instrument development informed by extremophile physiology. For example, sensors tuned to membrane lipid classes found in acidophiles or halophiles.
That is a great suggestion. I will add how knowledge of extremophile membrane chemistry could inform targeted mass spectrometry protocols and extraction methods that avoid destroying key biomarkers.
One last thing. When you write about analog sites, be explicit about why each site is analogous to a target body. For instance, Rio Tinto is an analog for acidic, iron-rich environments on Mars, and hydrothermal vents are analogues for ocean worlds like Enceladus and Europa.
I will make those links explicit. Thank you, Dr. Harris. This really helps me structure the argument that studying extremophiles refines both what we look for and how we build instruments to look.
You are welcome. Bring a draft and we will review the evidence table and the instrument ranking before you submit.
Listen again to part of the conversation, then answer the question.
Make sure you explain why multiple lines of evidence are necessary for declaring detection. A single organic molecule can be produced abiotically in a variety of chemical reactions.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.